Hey all, welcome to Parker's Reefs. On today's episode, we're going to use the ReefLabs P3 test kit to do a DNA test on the Dream Reef tank. Alright, thank you for joining me on yet another episode of Parker's Reefs, and as touched on in the intro, today we are going to get our nerd on. I have seen advertised on some of the Facebook pages around the reefing groups here in Australia, a brand new company by the name of ReefLabs advertising their P3 DNA test kit. Now, I have one in my hands right here, and the P3 component stands for pests, parasites and pathogens, and basically what this kit does includes all of the tools required for us to get a quality DNA sample, includes free express return shipping. It also obviously includes the process at the lab itself, which is here in Australia, and then gets put through the algorithm to provide a comprehensive report. So I figured today we'll go through the process. I'll show you what you get in the kit, how you go about taking that sample. Then what we're going to do is actually take a video call with Dr. Tom, the man behind ReefLabs himself, and get him to personally go through the results that I got in my kit. So without any further ado, let's roll the footage. All right, a number of weeks ago now, I received my P3 test kit here from ReefLabs, and I figured the first thing we'd do is open it up, go through all of the items you get, because you do receive everything you need to collect your sample and even the return shipping back to them. So it's quite a comprehensive kit, and there are a few steps involved in the process, so I figured we'd go through it here. Firstly, there are some instructions, this very detailed sheet here with a QR code up the top there, which will take you to the videos that show you what to do. I'm going to go through the process here, but I recommend you checking it out yourself as well. Firstly, you need to wash your hands, give them a thorough wash. We don't want to contaminate any samples at all, so giving them a good wash and dry there before jumping back to our kit and putting on our included sterile gloves. So I'm going to do that now. I've got my hands nice and clean, and I'm going to get my gloves on there just so I don't get any of my dirty hands or oils or any sort of thing that's on my hands that's not in the reef tank on the syringe or any of the sample products at all. So now that we've got those on, we're right to proceed with the rest of the test kit. Now you can see I've got a number of products in front of me here. This one here is of particular interest. This is the filter, and it does have a number on there, which is what I need to register on the website. So the instructions recommend I write that down on this piece of paper here so that when I post everything back, I have that number there and don't forget about it. Now, what I need to do is grab this 50 milliliter syringe here. I'm going to open that up, and we're going to grab some water from my tank and pass it through this little filter. So you can see here, head on over to my tank, and I've got my syringe. Going to drag 50 milliliters of water out of the tank with that, and then I'm going to take it back over and pass it through. You see 50 mil there, pass it through this filter here. Now this is a very specific and specialized filter that allows water through, but it actually collects the DNA as it goes, which is super unique and interesting. It does have this directional option on it here, and it does have a screw-on attachment to that syringe. So I'm just going to screw that on there, grab my cup of water, because as I said, the water will pass through it, and I'm going to pass 50 mil of water through this. Now the instructions do say that we need to do this a number of times. So I'm going to do the first one at real speed just so you can see that it is a fairly resistive little filter. The water doesn't flow through it very quickly. You need to push fairly hard, and you can see that water coming through there. Now, hopefully no one needs to go to the bathroom, otherwise you're probably going to have to pause this video and take care of that. But in the meantime, I'm slowly pushing this 50 mil of water through into my cup there, making sure that I don't contaminate the sample at all. And as you can see, it does take a little while. I will do this another 3 to 4 times. In fact, I'll do it 4 times total to give me a total of 200 milliliters. The instructions do recommend you do it up to 6 times if you need to pass enough water through there to make sure you get a quality sample. And I must say, it doesn't hurt to pass it through a few times. So you can see here, it takes a little while. That's okay, we're nearly there. The next few ones, I will speed up the process very quickly so you don't need to see how long they take. But it is something you want to take a little bit of care of. These test kits, they do come at a little bit of a cost, so you don't want to contaminate your sample or damage it or anything like that. Follow the steps very slowly and carefully to make sure you get things right. But now that I have got 200 ml of water through my sample, I'm ready to progress on to the next step. And it does say here that I need to write on my instructions how much water I've passed through the sample. So I'm just putting 200 down there. Now, looking at the next steps there, I need to actually draw some air into my syringe and pass that through the filter to make sure there's no excess aquarium water sitting in the filter. And you do actually get a little instruction, or a little diagram I should say, on the instructions to show you how much is too much water and how much is acceptable. So I'm going to pass the air in through there. It does say to do about 30 milliliters of air connected to the filter, and while holding it vertically pointing down, press the syringe plunger to expel the water from the filter. I've done that now, well, I'm slowly doing that now, and I'm fairly content with how that's looking, so I'm good to proceed on to the next step, which basically means I need to screw off this big 50 ml syringe and get this fantastic preservative on there instead. Now, this is an interesting little thing. I must admit, I am no science lab expert, but this sort of makes sense to me. We've got a live DNA in here. We want to make sure that it stays intact and preserved before it gets to the lab. So we're going to put this little syringe on here, which is the preservative, and then we're good to start squeezing that into the filter itself so that it is nicely preserved. Once you have done that, you'll see that little pink cap that came on that syringe. Pop that on the other side so you don't get any exposure to air or any other sort of contaminants. Put that back into the bag that has the barcode on it, and you're good to send that off. You can see here, this is my kit completed. It's got the syringe on one side, the cap on the other, the preservative, and obviously my water sample in there. I can get that all prepared, put that in the bag, and then send that off. Just before posting it off though, there's one more step you need to do. You obviously need to register on their website so they know whose sample this is. So just quickly now, I'm going to jump onto the reeflabs.com.au website, which is of course where you can buy their products. Going to log in or create an account here. Just going to skip over the parts where it has my personal information, so don't get any stage 5 clingers out there reaching out. But essentially just quickly go through a little bit of information about your aquarium, any issues you know that are in the aquarium there, any other sort of information you can provide to the ReefLabs team to help them really deep dive into what's going on in your tank. Then it's just a matter of waiting a couple of weeks until your results come in an email. But for us, we're going to jump on a call with Dr. Tom to have a deep dive into the ReefLabs experience. All right, here we are. I've been super fortunate enough to get an extra level of service that you don't normally get with the ReefLabs experience, but fortunately for me and for all of our viewers, we've been able to get Dr. Tom on the phone here so that he can take us through everything that is ReefLabs and even have a little bit of a deep dive into the P3 results that we got from the Dream Reef tank. So firstly, Dr. Tom, thank you so much for taking the time. I really appreciate it.
Thank you for having me. I'm excited to be here and discuss your reports.
Yeah, fantastic. Maybe before we jump into the reports, can you give us a little bit of a background onto how you got into this space and creating ReefLabs and just the journey so far?
So I've been in the marine hobby. I've kept corals and marine tanks for, I would say, 17 years, again, since I was a teenager.
Yeah.
Always had tanks. Then went into my degrees, did biotech, did a PhD in aquaculture biotechnology.
Nice.
Worked in industry for a bit in a similar field, and then wanted to do something that I'm passionate about.
Yeah.
It started with issues in my tank and friends' tanks. So we use this technology, DNA sequencing, in academia, in research environments quite often. So why not apply it to my reef to find the problem?
Yeah, you bet.
And it definitely illuminates what's happening, because diseases, microbes, we can't really see it. Yet if we use certain technologies, we can amplify and actually get the data and understand it better.
I mean, it comes into that space, of sort of the old school reefing technique where, you only dose elements that you can test for and things like that. But then the whole pest, pathogens, bacteria, things like that, we've not been able to test for. So it's been this huge question mark in the hobby for quite some time. And realistically, we'd only wait until we saw the after effects and then have a slightly educated guess as to what could be causing that, and then fairly blindly treat from there. The space you're in completely changes that.
Yeah, so a reef environment is very complex. So especially in a tank where the volume is very small, it's very dynamic. One nutrient goes up, it affects the biology. The base is to keep the chemistry consistent.
Okay.
This is before any other testing. You always need to focus on the chemistry.
Yes.
Because that's the baseline of the marine ecosystem. So ICP, I definitely support it. I test ICP myself. It definitely gives you another layer of understanding your water chemistry. But then you have the biology, which is also very dynamic. It also fluctuates and changes. And DNA testing can also give you answers on how your tank does biologically.
Interesting. Very interesting. And I know I've done a report and we will have a look into it, but I'm assuming there's all sorts, and I don't expect you to give away any sort of industry secrets or anything like that, but it seems like a big, like I understand you've done the study and you've worked in the field and that, and you've had reef tanks, but for the average aquarist like myself, it seems like such a big leap to come into a space like this and apply it into reef tanks. This must have taken you some time to really get a handle on things.
It was definitely a process, and again, it's still evolving.
Okay.
We do have a lot of data on from other researchers. For example, we know that Vibrio fortis is a fish pathogen, or we know Acropora eating flatworms, coral pests. Some of the data is available, so we've compiled into a database.
Yes.
So we have our own database of what we're interested in. So pests, pathogens, parasites, toxic algae as well, which we'll discuss, and also microbiome testing, which looks at bacteria and archaea.
Yes.
So assembling a database takes a long time. Verifying the technology, seeing the limit of detection. So it's always evolving. We're always going. We're actually going to stores and ask them if they have this pest or DLC coral. We actually buy corals that we can see pests on, so we can sample it and add it to the database.
I bet that got some strange looks from store owners when you went in asking for that.
A little bit, but I explained to them what we do, so then they're happy, happy to help us. Always evolving. The more data we have, the better we understand it. So we always look at the data. We see, hey, we have this tank or a collection of tanks that are looking good. Let's see the microbial population or the ciliate population, because we don't really know too much about, for example, ciliates, but we can see them in our DNA tests. Let's compare that to tanks that are doing bad. Can we see any ciliates that are in the bad tank and are not in the... So make associations, correlations to improve the reports and our data analysis. So it takes time. And it's always growing, it's evolving, and it's becoming better and better with every test we do.
Incredible. Is there any chance you can give me a very, very high-level overview into the process that goes into doing one of these tests?
As a customer or as a lab?
As in, on your side in the lab. I've shown in the video what I need to do on my end, and they're grabbing the sample and how what the steps you need to do there. But then once you receive the sample, and I understand that it might not be possible to do that at a very high level, but a real basic overview of the process.
For sure. Let me show you. I'll just share a slide if that's okay.
Sure. Okay, nice.
Okay, so it is a complex procedure. It involves a lot of lab work, a lot of hands-on work, and machinery. And then you have the analysis part of it. So let's take it step by step.
Sure.
This is a very simplified version of what's actually happening.
This looks right about my level. It's good.
Sam, if you have any questions, feel free to stop me, and I'll
of course.
I'll go over a particular. Okay, so the customer gets a kit that has everything you need to sample a DNA from your tank and strip it to our lab in a stable manner so the DNA doesn't degrade.
Yes.
So we receive a mixture of water with DNA with microbes and other gunk. We don't want anything else. We just want the DNA. So we extract and purify the DNA. Then we have pretty much genomic DNA from dozens or even hundreds of different species.
Mm-hmm.
And to identify this, to speciate them, to identify the species, we're looking at a very particular part of their genome. So it's called a genetic marker.
Okay.
So, for example, in step 4, that could be a long piece of DNA with 10 different genes, or it can be, let's simplify it. We have a piece of genomic DNA, it has many, many genes. We're only focusing on one of them. It's called a genetic marker.
Yes.
The reason we're using a genetic marker is because it's very conserved. So, for example, for bacteria, for the microbiome test, we're using a 16S gene,
Okay.
which all bacteria and archaea have. So when we amplify that, we capture all the microbial population, all the prokaryotes, all the bacteria and archaea. Now, although it's conserved, it does have differences. So, for example, bacteria A will have some differences in the code compared to bacteria B, and we can pick that up when we sequence it. So, for the P3, we're looking at eukaryotes. So they're not bacteria, they're a bit more evolved. That includes animals, that includes microalgae, invertebrates like corals, ciliates, etc., etc. So we get the gene marker, we amplify it. So we only select that part of the genome.
Okay.
And we add adapters. So this is the light blue DNA.
Yes.
And these adapters, so each sample that we get gets different adapters, and that helps us identify which sample it came from. Because eventually we're going to sequence it all together. After we prepare the DNA, it's called a DNA library.
Okay.
We run it in the sequencing machine. You leave it for a few days to do its thing, and then it spits out a long list. So a long list of genes. For each sample that we receive, we sequence 50,000 different pieces of DNA to get a higher coverage. The data, I can show you the data, the raw data, what it looks like. It's just a long piece of code.
Can imagine.
With just random, it's not random, but just A, T, C's, and G's.
Sure.
Which, with the human eye, you cannot know what it means. There's no way for you to differentiate. It's just too complex. So we use computers for that. It's called bioinformatics. So we use computers to help us analyze DNA, protein, etc. The raw data goes into our algorithm. That does all the magic. It looks at how gene A that we find in our sample matches to our database. And then it gets an ID. So then we're able to associate the sequences that we've sequenced to a database and get a long list of species. So we get the ID, but we also get the abundance. How many DNA of bacteria A were detected?
Right.
So this is how we can also come up with the numeration or the quantification of the bacteria to give you bacteria or pests, parasites, and pathogens. So this is a short story of how DNA sequencing is done. I can get into more details if you want.
No, that's sufficiently over my head as it is. It's incredible. From a very short recap from a layman's perspective, I can see there's some serious steps involved there, from obviously even just the preparation of the sample and maintaining the DNA in that sample so that it comes from my tank to you and the lab. Then there's processes involved to get the pure DNA out, to then identify them and amplify them, run them through some incredibly specialized machinery, and then get to an even more specialized algorithm to make sense of all that data, and then use your knowledge and your experience, the data you have, to then produce these nice reports that the end hobbyist receives, which gives them a good indication of what's going on in their tank. All right.
Would you like to go over your report?
Yeah. So, a little while ago now, I sent in a sample of water so we could do the P3 test report, and I was fortunate enough. You also said that you'd be able to run the microbiome report on the sample as well. So I've been able to get two sets of information from the Dream Reef tank, which is fantastic. It's good to point out that there wasn't any. There wasn't a critical need. I wasn't experiencing major deaths or issues or anything in the tank. I just saw through online advertising this new ReefLabs P3 test kit available, and I reached out and said, look, I really want to do a test on the Dream Reef tank just to see where things are at and to experience this technology and share it with the viewers out there. Now, what I'll do is I'll share on screen the report that I've got. So I'll share the P3 report, which is our first one. All right, so this is the PDF that gets sent out to the hobbyist once we've gone through when you and your team have gone through all of that process, put it all through that specialized equipment, specialized processes, wealth of data, wealth of algorithms. We then get a report, which is the P3 test report, and it's called P3 because it tests for pests, pathogens, and parasites. Is that correct?
Correct.
Excellent.
Also, we do report toxic algae as well.
Yes, yep. But it kind of takes away.
It just doesn't have a P, so
It takes away from that catchy name. So it's maybe P3 plus. Maybe that could work. I'm not sure, but we'll roll on through, and you can see we get a nice little introduction to the report here, and it does say that it tests for pests, parasites, pathogens, and toxic algae there. Explains some of that DNA sequencing that we talked about before, and then tells us the information that we're going to get with the sample overview, the threats detected, the recommended actions and preventions, which I think is super, super important, because it's one thing to be sort of told there might be traces or high levels of a particular DNA sequence in your tank, but then what you do with that information is really the next step. And to get that recommended actions and preventions, I think, is very, very important. And then, of course, you get a full list of species, which is sort of the hobbyist's view of the full set of data, which I think is really, really good. Now, we jump into a sample overview here, and you can see that there are a number of 43,200 DNA strands sequenced, and then you get a number of diversity, so the total unique species detected, and this sample here is 219, and then the threats detected as 4, and you get a nice little graphical overview, I'll say, of that, where you can see like stony corals, sponges, dinoflagellates, sea anemones, bony fishes, fungias, things like that, all in the sample there, and that just gives you a nice little visual representation. When we scroll down, we get a nice little aquarium overview, which does put in plain English, well, plain enough English, what's found in that water sample there. So in mine, it says that there's obviously acro, there's Bacilloporas, Montes, Ceratopora, Micromusa, and some other coral types in there. There are some beneficial microalgae in there, which is important. And there's some DNA from sponges as well, which again, super important in the ecosystem of a reef tank. There's some other things in there like turbos, trochus, and snails, and other invertebrates like that. We've got some, I've forgotten how we pronounce this, ciliates, was it?
Ciliates.
Ciliates, my apologies. And can you tell me what sort of they are? What?
So ciliates are eukaryotes. They're not bacteria. Okay. They're a bit more higher up on the evolutionary tree. Right. They're single cell. They're single cells. Quite small. You cannot see them with a naked eye. They do have a very important function in aquariums and the ecosystem. Mm-hmm. Mainly, they feed off detritus. Okay. Some feed off other bacteria, and they're found everywhere in the environment, especially marine. So marine, freshwater. So it's a good sign that we see ciliates. Yes. Some of them have been thought to be pathogenic to corals. There are some species. However, later we found that they do, as they're detritivores, so they eat decaying and dying matter, they are associated with dying coral. But most likely it died because of a bacterial infection.
Yes.
just coincidentally they were also there. But psyllids are a normal part of the biome of the tank.
Okay, good to know. So they are something that, they're obviously going to be there, but you want to have a reasonable diversity of them and in sort of stable numbers, I guess, rather than, one type dominating over another type or something like that.
So currently we don't have the data of saying which one is better to have in higher amounts and which one is not. What we do want to see is a diverse community of them.
Yes.
So, for example, if I see in the relative abundance figure, if I see a higher abundance of ciliates, that would be a red flag. You have a lot of ciliates. That could mean you have maybe it was a high def, ciliate population overgrew and they're overtaking other organisms.
Okay.
In your case, you had a very good and healthy levels of ciliates.
Good.
In your tank.
Good to know. I feel like I'm at the general practitioner's getting a health report, but I guess in a way the tank is an extension of me, so that's always good to know. We then jump into just again in the sort of the plain English or language overview, a quick mention of pests to say that there were low to medium levels of aptasia, which is something that I'm well aware of. There's a low level of vermitid snails, which I'm also aware of. And that there's a low presence of these, which is nothing to be too concerned about, but good to know nonetheless.
I just want to clarify that the low to medium levels represent the DNA, so the abundance of the DNA.
Yes.
It doesn't necessarily represent the number of individual animals in the system.
Yes, good point.
So we're just doing a correlation. We know that tanks that have up to this percent don't have a full outbreak of aptasia. So there maybe some differences, but everything referred to here is DNA levels. So low to medium levels of.
Very good point. And then there is also a flag for monitoring here to say that a particular type of dinoflagellates was detected, but it was detected in low volumes, and it doesn't suggest an outbreak, which is correct. I don't have an outbreak, but I have seen low levels of dinoflagellates before, and it gets into that sort of point where you're never sure if it's a cyanobacteria or a dino. Thankfully for me, it didn't take over, which indicated that if it was a dino, it definitely was at low volume. So interesting to see that you're able to find that there, and that you give some information and some recommendations of what to look out for, and things like nutrients, keeping them above zero, running some fresh activated carbon, running UV, and also to mechanically filter the sand through a fairly fine filter sock. Increase flow and micrograzer diversity, as well as performing water changes. All good common sense if you are seeing some sort of dino there and you want to tackle that.
Yeah, so this particular part is in case you are seeing a higher number of dinoflagellates.
Yes, makes sense. And then things are just wrapped up there with overall the aquarium suggests a functioning reef system with a good biodiversity. There's no major red flags. There's some minor nuisance organisms at trace levels, and just to keep monitoring and good housekeeping, retest if symptoms appear to get worse, which makes good sense to me. Moving on from that sort of high-level overview there, we then get into a little bit more detail again. I love the way you sort of just unwrap the next layer and you get some more information here. So we jump into a little section for threats detected in the aquarium, and that these ones, there's a paragraph there which explains sort of how they come through in the DNA sample, and higher percentages, as you touched on before, could suggest that the organism's more active. And you've gone into some detail here around some aptasia, some low to medium levels of aptasia, and an actual percentage in there, which is interesting. So for anyone playing along at home that has a test done as well, they can compare the percentages they got with mine, which will be interesting. Immediate control is advised, which for those who have had experience with aptasia before know that if they're in there, they can quickly take over and get quite out of hand. So I definitely do have some natural solutions in there in Australian stripies and some other fish that just like to keep them at bay, which is fantastic. And you've also got a guide for effective treatment options here, which you can follow through and get even more information on your website, which is fantastic as well. We do jump into the next one there, which is, I'm going to struggle to pronounce that, amphibian.
Amphidinium.
I'm 80% of the way there. Now, tell me about these ones. This one, I've got an acceptable low level. What are we looking at here? What's this sort of threat in the system?
So, Amphidinium, they are dinoflagellates. So,
Yes.
dinoflagellates are a normal part of the reef ecosystem.
Of course.
They're everywhere. Some species are known to produce toxins.
Okay.
Some of them affect fish, some of them affect corals, other invertebrates, even us.
Yes.
The reason why we flag it is to alert the customer that you have this species in your tank. Mm-hmm. Be mindful of your tank conditions and try to prevent a bloom.
Sure.
Once they take hold, it can be a bit tricky to control them.
Yes.
And you can lose animals. Yeah,
it can be quite aggressive. Yeah.
So if we look at, if we go back to your species relative abundance.
Yes.
Chart.
Yes.
You will see that there is dinoflagellates in the yellow.
Yes.
But that's not 0.1%. So how come I have such a... So we show you the ones that are known to produce toxins. However, most reef tanks, the predominant dinoflagellate is a symbiodinium. So this is the symbiotic dinoflagellate that lives in coral tissue and is very important.
Gives us the color.
And the coral is constantly regulating how much dinoflagellate is in its tissue. Mm-hmm. In response to lighter environment, so there's always some in the water. So the majority of the dinoflagellate that we see in reef tanks are actually symbiodinium, and they're the good ones.
Okay.
That's why the block for dinoflagellate is a bit bigger than the 0.015% of amphidinium that we detected. Just to clarify
that makes sense, and thanks for pointing that out. You do also, again, and I appreciate you've got a link to for a guide for control and mitigation strategies there, which is excellent information. Again, I pointed out that it's one thing to test to see what's in your water, but then once you find out what is in your water or what DNA is existing in your water, what do you do next if you feel it is taking hold or going in a direction you don't want? That's very, very important. The next one we've got here is a golden algae, and I'm going to go with the layman's term, their golden algae, because Primesium parvum, I'm not sure if I'm remotely close on that, but golden algae I'm comfortable in pronouncing. This one came in at 0.01%, and you have mentioned that is a normal level in a diverse system, which is fantastic. But then it does scare me a little bit, and I read the next bits about the toxins can have harmful effects on aquatic organisms, all sorts of things there, and killing cells. It sounds like an incredibly dangerous thing. I understand small trace levels, completely normal, expected to be there, but have you seen many systems that have higher levels of this golden algae in there?
Yes, so I have seen systems that have over 5%.
Wow.
And that is definitely a sign of a bloom.
Yes.
And we notify the customer that they need to act now.
Yeah.
For example, in aquaculture or even offshore aquaculture fisheries,
yes.
Natural blooms, sometimes they do bloom naturally. Mm-hmm. And as the current brings them on into the fish cages, there is mass fish die-off.
Right.
So this is where we get the data from. So we see what we know about these species. Can they harm our fish, our corals, and we add it to the list. Now they're not inherently bad. They're bad when they're overpopulated.
Yes. Like anything, everything is fine in moderation. When you have an outbreak of something, that's when things go bad. Very, very interesting. On to the next one there, vermitid snails. I'm definitely aware of some of these in my tank. And targeted removal, I was going to say, do you have a solution for vermitid snails? Because we're in the hobby, I'm not familiar with, at least in Australia. I know in some other parts of the world they have some natural predators that can go after vermitid snails. In Australia, we're still really only looking at manual removal, and it seems that's your experience as well.
So we are working, I think the team maybe have released it already,
Okay,
a protocol on how to eradicate them.
nice.
Again, there is biological, there is environmental, and then I wouldn't use any chemicals because they're a gastropod, they're a snail. There isn't really a safe chemical treatment to target them but not harm
Yes.
other invertebrates in the tank. But we definitely found that if, like, when we feed a lot of small particulate food, the population just increases. There's mass spawning, and then they spread everywhere.
Wow.
So feeding fine particulate food definitely boosts their numbers and reproduction capacity. So in some tanks, if you don't need to broadcast feed, and you do deal with a lot of, or you see some signs of vermitids, maybe knock it down a bit.
Sure.
When you feed heavily, you can definitely see the mucus strands because they're filter feeders. They
You
send a
definitely
long
can. Yeah, you'll see those cobwebs go everywhere.
Yeah. So the issue is when you have too many of them, it can irritate corals. So they're not a high level threat. They're more of a nuisance and potentially a cosmetic issue as well.
Yeah, of course, yeah. There's nothing worse than if you're working on the tank and the shells on the vermitid snail, that little tube, is so sharp. If you happen to cut yourself on that, it gives a pretty nice cut and will bleed pretty badly. So they're best avoided, but good to know that a potential treatment may be on the way, but in the meantime, targeted removal best. And as you say, if you wanted to stop the numbers exploding, maybe you avoid broadcast feeding where possible.
Yeah. So just to clarify, we don't have a solution for them, but we compiled the available evidence.
Yes.
A lot of it is anecdotal evidence.
Yes.
For the users to follow and see, hey, what can I do? So, for example, reduce feeding. There is some people choose to manually remove them, but you need to make sure you remove them from the base because breaking the tip will do nothing. They will retract into the base and then just regrow it again. Dipping doesn't seem to do much. They haven't sort of like a barrier on a perculum, so they can shut their door, and then when the dip solution is out of the way, they come out. There's one thing that we saw that people use, again, it's anecdotally, we never actually tested it scientifically, is to use calcium carbonate powder.
Yes.
The rationale is they can't digest it, they filter feed, they're going to catch it as it's going to clog their intestines, they won't be able to process food, and they will die. So this is the rationale behind it. We will post the page about the eradication plan, if it's not up already, for viewers to go online and have a look at
follow along. Yeah, no, good to know. It's definitely a challenging component out there at the moment, but hopefully we're edging closer towards some, even if anecdotal at this stage, some solutions that may well be worth trying. And at least with calcium carbonate, it's not a huge downside. I mean, unless you're going ridiculous with the stuff, there's little risk to be had. So if it works, great. If it doesn't, it's probably not a lot lost.
Everything we put in our tank would have some sort of effect.
Yes.
So you're right, calcium carbonate is benign. However, it needs to be tested, but it would be possible that it might precipitate some elements on itself.
Yes. Yeah.
So again, it's a low risk method, but it could have some sort of side effects.
Low risk is not no risk.
Yeah.
heading back to the P3 report, we then get to a section where we've got a full list of species and whether they were detected or not detected. And when I scroll down, it starts off with pathogens, gives you a little explanation as to what pathogens are, and then we get the full list of things that the P3 test looks for, and of course it gives a little the common name, the description, the actual scientific species, and whether it was detected or not detected, as people can see on screen here now, these first few not detected, which is definitely a relief. And you can scroll through all of those ones there, and you can see in this pathogen section all of mine were not detected, which is a good relief, particularly things like marine velvet, marine ick, uranema. There's some pretty nasty things in there that you ideally don't want to come back in the results, but it is kind of the point, or one of the purposes of this test kit here is if you've got fish, you've got them in a quarantine or something like that before you go releasing them into your Pride and Joy reef tank, you can do a test like this and see if something like uranema or velvet is existing in there, particularly something like uranema that might sort of hang around for a while before it rears its head. People can test that, get the results, and then make a decision based on that. Heading down onto the parasite section, and again we get a description of the parasites, and same process applies here where you get to see what the host of the parasite is, what the common name is, description of that, and then all the different types of species, and even within the species you've then got in brackets multiples of those species. So it's an incredibly in-depth report. Thankfully, again, most of these, if not all, I'm scrolling, got quite a few pages of them, no parasites detected in my sample, which is it's a good result for me. Another big wipe of the brow, very happy with that. Heading on down into pests, and again, same process again. This is the third of the P's. We get the description, down into the common name, description, the species, and the results. And I can say here that as we saw in the overview above, some things like one species of the vermitid snails was detected. Likewise, one species of the Aptasia was detected. Looks like all of the other pests there in my list not detected, which again, massive relief. There are some things that you definitely don't want to see, like boring bivalves, clamworms, fireworms are another fun one. And it's, again, this report's not necessarily to say that they don't exist in your aquarium, but the DNA was not present in the water sample, which means that they're probably not going to be in any sort of abundance. Is that correct?
100%. So, for example, so fireworms we can detect. So, not to mistaken any polychaetes like bristleworms to fireworms.
Yes.
We specifically put fireworms in there, the ones that are tend to be more carnivorous,
Sure.
tend to potentially eat prey on fish, those that grow to a larger size than our normal bristleworms.
Yes, yeah, definitely.
For example, the boring bivalve.
Yes.
They are extremely hard to detect as they are mainly inside, deep into rock structure or deep into the skeleton. So we've only detected them once at very low level. So to me, it seems like the DNA, it's hard for the DNA to get out into the water column for detection.
Yep.
If, for example, you take the base of the coral, we sample that, there's higher chance of us detecting it. But you're right, through the water is a dilution factor.
Yes.
On top of that, there's less access, less water flow to carry the DNA out. So we don't really see it very often, although they may be there.
Yeah, of course. Very interesting. And then to jump into that plus part of what I'm just now calling the P3 plus result, where we get to the harmful algae alert section. And again, we get a nice little overview as to things that can potentially be a risk to fish, corals, and invertebrates. And then we jump into the genus, the toxins produced, the effects, and the species. And I've got, I was going to say a couple. I do have a couple. I've got a couple of algae types detected there. oh, geez, this we test my nomenclature, but I'm going to say Amphitinum operculatum has been detected in my tank, and this is an interesting one. This is an algae that can have toxins that can damage fish gills, induce systemic stress, immune suppression. The toxins can harm fish, invertebrates, and zooplankton. So it's a serious algae that I probably want to keep an eye out for and have a look at what I can do about that. But any further insights into that on what I should be looking out for?
Luckily for you, it is a scary algae, or the description is scary. But luckily for you, the levels are very low. I don't suspect any harm from that. Yeah. It's a harmful algae alert. Just know that it's there.
Sure.
If you have a dino bloom, then you know what might be the main culprit, or what kind of species might be there.
Oh, of course. So this was the strain of the dino is further down, isn't it? Yeah. Okay. Yeah.
The toxic algae are broken down into not all toxic algae are dinoflagellates.
Yes.
The Amphidinium is, and then you have another one, which is a haptophyte.
Yes, I do too. Yes. Tell me about this one.
It's not a dinoflagellate.
Mm-hmm.
But the way you treat for it, or the conditions that make it bloom, are very similar.
Okay.
So in our in the information page about dinoflagellates, we put them together. We state that they're not the same thing, but the treatment is the same.
Yeah. Okay. So
we tend to see that in very low nutrient systems, they tend to take over.
Right.
So they have a few extra tricks up their sleeves to make him extract more nutrients from competing algae. They've got enzymes. They have more of a protist than an actual algae.
Right.
They have enzymes that can break down organic carbon for energy. They have the ability to use organic phosphates, organic nitrogen that we don't really test for. So we might have low phosphates, but inorganic phosphates, or elemental, or the simple inorganic phosphates, but we might have a lot of organic phosphates in detritus, on the rock surface, on biofilms, and they can tap into that.
Right.
So very low nutrients, they have the upper hand compared to other types, and enables them pretty much to overpopulate.
Yeah. Right.
So zero nutrients are not good.
No. Thankfully, the hobby's moved away from that zero nutrient, ultra low nutrient phase. We're now into the world of sort of 5, 10, 15 plus of nitrates and a decent balanced amount of phosphate, which is definitely seems to be producing better reef tanks than we were having, say, 15 years ago. So I think we're on the right path there. All in all, to wrap up that P3 report, I mean, I've only ever seen one of these before. I'm assuming you've seen dozens, if not hundreds of them. I think that's not a bad result for a tank that's around 5 years old now. It's definitely not an Instagram tank where it has never had a problem at all. It's gone through its share of pests, its share of uglies and things. But, I mean, tell me if I'm wrong, but looking at the report, I get the impression that things are looking pretty good there.
100%. I guess you're likely just to have a few vermitids and some aptasia. We've had some tanks that had worse things and that are much harder to deal with. So I think your tank is, according to this report, is doing really good.
Great. Good to know. Definite weight off my mind. And like I said, I think the majority of the time when people are getting these tests done, they're looking for an answer to a problem they're currently experiencing, where I was just a little bit more curious and just wanted to see where my tank was at that point in time, which is, I guess, also an equal, it gives people a good baseline, I guess. They know where things are at before, they can see where things are at now when the tank's doing well, and then should they experience problems in the future, they can do another test and see if things have changed from there. So I guess it's not completely unique to be in that situation, but good to see nonetheless. Should I jump over to the microbiome report, which is a new, it's a beta report that is something just new that you guys are working on? Is that correct?
So when we did your report, it was in the beta stages.
Okay.
Now it's out, and customers have been sending in the tests.
Yeah, great.
I did actually find some mistakes, or mainly spelling mistakes, in the report that I sent you during the beta testing.
That's okay.
So we'll ignore that part.
I'm definitely not the spelling or grammar police, so don't worry about that at all. For me, it's more about the information that's in there, and I'll just share that on screen now, and I'll scroll through. Again, it goes in a very similar sort of fashion to the P3. Actually, well, yep. Onto that report where you get that nice little overview, the things we're going to look for, things we're going to find, and then we get onto the information specifically around the microbiome balance, which is definitely a part where I think that we're seeing the hobby go towards, thanks to people like yourself that make this sort of technology available to us, where we're often curious around that microbiome and what sort of types we have in the tank, what diversity, what volumes, what balance we have. If there's low numbers, high numbers, it's all interesting. And as mentioned here, a low diversity or a skewed abundance of a particular microbe can be represented in some stability issues and things like that. So it's an interesting report. Now, I've scrolled down to the balance report here, and it's, I mean, to be fair, even for someone like myself who is not in this space at all, this makes sense to me. We've got a nice color bar chart here where you've got the microbial diversity gauge. You've got that separated out into low, moderate, high, and very high. Mine looks to be somewhere in the sort of just above middle of very high, which is for bacterial species count, which I think's got to be a good thing. And you've got some wording around there to say that it looks like it is a healthy tank, and the sample contains very high diversity levels of 681 species of bacteria and archaea. Am I pronouncing that right?
Just to correct you, a high diversity doesn't necessarily mean healthy tank.
Okay, good. Yeah, tell me.
Because I can have high diversity of bacteria, but then also have some pathogenic bacteria.
Yeah, good point. Okay.
What it does tell me, so let's break it down simply. So
Sure.
microbiome is extremely dynamic. So in this report, we really tried to simplify the complex data that we have, the interpretation. As you scroll down, we made figures for categories.
Yeah.
Here you can see a color change. Red probably bad, and it's true low diversity is not healthy microbiome.
No.
High diversity is better, and the reason is microbes in the tank are at war. They're competing for habitat. They're competing for nutrients. If it's cyanobacteria, it's competing for light. Yes. Some of them live together, help each other. Some of them produce compounds that inhibit other. So it's a constant war of territory and nutrients. This is a snapshot to visually see which microbes or which families dominate my tank. So it's easy to see the abundance and diversity. So we can go over your results if you want.
Sure, yeah, that'd be great, because I must admit there's a lot of big words in here, and I can see there's a lot of data and a lot of interesting things here, but I just can't quite digest it yet. So yeah, give me some more information. Let's go through it.
So just by looking at this, it can tell me a few things about your tank. So Altraumonadaceae, it's a bit of a mouthful word. They're very common bacteria in marine ecosystems. A very wide distribution. They do. So their main fuel source is dissolved nutrients, sugars, amino acids. It's normal to have them in high number, like your tank. But your tank maybe just has a bit extra.
Sure. Yeah, it looks like. I mean, at a quick estimate, there, it's about a sixth of the volume is in that group. So it's a pretty high concentration.
abundance. Yeah, and then we go down to Rhodobacteriaceae.
Yes.
They are mainly associated with biofilms or surfaces. Again, very common,
Okay.
very important, and your levels are not extremely high. They're very healthy levels.
Okay.
Um, they also feed on organics.
Okay.
So, for example, a lot of waste degraders are from Rhodobacteriaceae family. High DOC would increase their population. Um, very important group for waste management, but it also
Yes.
it may imply that at least in terms of the DOC in your tank might be slightly higher.
Okay.
And then the other higher group is Vibrionaceae.
Yes.
So Vibrionaceae is a family where Vibrio is found. So Vibrio,
Yes.
some Vibrios are problematic slash pathogenic.
Hmm. Yes.
Vibrionaceae, not all members of Vibrionaceae are bad. A lot of them
Okay.
are normal part of the microbiome. Some of them are even good. Again, they also degrade waste. They're important for nutrient cycling and sort of waste recycling of your tank. So
Okay.
three dominant groups that rely on DOC or high amounts of waste. So to me, just looking at
Yes.
this, it tells me that either you feed a lot or you have a high stocked tank.
Okay.
Now, another thing that pops out is the lack of Pelagibacteriaceae.
Yes. Yes.
Flav-
That's noted there.
yeah,
Yeah, that it was not detected.
Flavobacteriaceae. Um, so Pelagibacteriaceae is a very interesting one. It's sort of like an indicator family. It's one of the so
Hmm.
it's the top one of the top three most abundant bacteria in the reef, specifically the Great Barrier Reef. If you sample the
Wow.
microbiome there, the water microbiome, you're going to
Okay.
see mainly Pelagibacteriaceae. Pelagic means in the water column. It's a planktonic bacteria.
Okay.
You don't really find it
Hmm.
on surface as much. It's mainly in the water column, and you find two species of cyanobacteria.
Okay.
One of them is Seniococcus. Um,
Right.
there are a few reasons why you don't have that. So,
Hmm.
healthy tanks or mature tanks do have Pelagibacteriaceae. Your
Yes.
tank doesn't.
Yeah. Yeah.
There is a few reasons why.
Okay.
They do like low nutrients.
Hmm.
Outer reef, Great Barrier Reef, low nutrient environment.
Sure.
They're slow growing, so if you do have higher nutrients, other bacteria will compete with it, and it'll reduce its population.
Sure. Sure.
Another reason, if it's a new tank, they're very slow to grow. Your tank,
Hmm.
I assume, is not new.
No. No.
And the third reason is using UV.
Hmm. Hmm.
So because
UV,
they
I
are,
do use, yeah, and as
imagine,
it's a water based, I imagine that would be quite susceptible to that.
because they're mainly found in the water,
Yeah.
um, the majority of the population would go through the UV, and they're sensitive
Yes.
to that.
Yes.
So, for example, if you stop using UV for 2 weeks or a month,
Hmm.
you should see their numbers come up.
Right. Right.
Now, although they're associated with healthy reefs, we
Hmm.
don't really know if they're crucial for healthy reefs.
Yeah. Okay. Yep.
So,
Yep. Yep.
because if you have it, it looks like a healthy reef, or it looks like the Great Barrier Reef.
Yes.
If I don't have them, it doesn't mean that my corals will die. It's not an
No.
indicator
No.
species.
Yes.
Are they beneficial? Do they have a benefit? Probably yes. We don't really know yet.
Sure. Sure.
Also, Flavobacteriaceae wasn't on the list or in the family composition.
Hmm.
Um, there are another specialized degraders, mainly polysaccharides like cellulose, um, agarose, proteins, and they have high association with macroalgae. So they grow macroalgae surfaces.
Okay.
For example, with tanks that have a lot of microalgae, whether it's nuisance algae or ketomorpha in the
Sure.
sun, you do tend to see a lot more or a visible amount of Photobacteriaceae.
Right.
How do you have nuisance algae in your tank?
I've got very low levels of nuisance algae. I do run a refugium, so I do have a macroalgae in there, but I do run UV sterilization as well.
Right.
So some things to look into
So
there.
yes,
Yeah, as you say, it's not
yeah.
necessarily
So
an indication that there's something wrong, but it is something to note that it's quite a common. It's quite a common bacteria. Is it a bacteria? No, it's a microbe.
yeah, it's a bacteria. Yeah.
Bacteria, yeah. So it's quite a common
Yeah.
bacteria, but it's not present in my aquarium. It doesn't necessarily mean that it needs to be successful, but it is interesting
Yeah.
nonetheless that it was not found in the sample. Yeah.
So it didn't make it to this figure. So we only include bacteria that are over 1% abundance. So it could be in the
Sure.
tank. It probably is in your tank because your tank is diverse.
Yes.
It's just dominated
Yeah.
by
Yeah.
other microbes that occupy the same niche. For example, the Rhodobacteriaceae. Um, Another thing I want to point out that is not a bacteria is the SOK, or the yellow band. That's the SOK P01 is an archaea. It's called the Asgard archaea. Um, these ones tend to live in anaerobic zones. Um,
Okay.
so again, they're waste degraders. They're important for nutrient cycling, and they're also able to uptake ammonia.
Right. Yeah. Yeah. Okay.
So they're also part of the ammonia or nitrogen cycle. And you have they're present in tank as well.
Yeah. Good. I assume that's a good thing. I mean, I don't. They're not in abundance there. They appear to be fairly balanced. So I'm assuming that's a good outcome.
Yes, it is. If so, I've seen some tanks that had a much higher abundance,
Hmm.
which to me, and I've written that in the reports for the customer.
Yes.
Um, it looks like you have a lot of anaerobic zones or dead pockets of flow
Right.
because they're anaerobes. Their levels will go high if you have a lot of anaerobic pockets,
Yes. Okay.
and it can be an issue
Yeah.
because then you can
Yeah.
have um, certain bacteria that can produce hydrogen sulfide and it's toxic.
Yes.
Um,
Yes.
so yeah, so your tank based on this, the microbiome looks healthy.
Yes.
Nothing really stands out. We'll look at the Vibrio because we don't really know what kind of Vibrio are there, but later in the
Yes.
report we should find the species if any pathogenic Vibrios were found. But
Okay.
overall, other than the two missing families, your tank's microbiome is diverse and it looks healthy and stable.
Okay. Okay. Good to know. Rolling on to pathogens, we'll have a look at the information there, and this is where we get an interesting table here of some pathogens like fish. We talk about Vibrio there, and a couple of different species of it there, and there's a total amount, and then the two different types there. Is this where we should be concerned looking at these?
You should be concerned based on the levels.
Okay.
Again, for example, you get a coral frag from a shop. Your tank is free of Vibrio. You bring in a new
Yes.
frag. That frag will have Vibrio on it, or it could
Yes.
have Vibrio.
Sure.
It's going to be in your tank. The issue is when they overpopulate.
Yes.
Most tanks that we sequenced so far have the family Vibrionaceae, and a lot of them did have pathogenic bacteria
Okay.
with varying levels of pathogens. So it's all
Right.
about the values. If your number is high, it means it's uh, potentially infecting corals or fish, depending on the species,
Yes.
and
Yes.
you need to watch out for conditions.
Right. Right. What
In
sort of
your
number
case,
are we talking about as that being a sort of tipping point for nothing to worry too much about? It's just something you should investigate a bit further.
It's very tricky to say. So if it's 1% per species, then I would probably flag it.
Sure.
Because a lot of these Vibrios are they're opportunistic pathogens. So at these levels, they don't really attack anything. They just do their own thing, eating detritus and organic matter. And then something happens that just triggers them to become pathogenic. So there's a thing called quorum sensing. It's bacterial communication. It can be from other bacteria. It can be some sort of environmental cue that triggers them to be virulent. We know that one of the environmental factors that triggers virulence is higher temperature.
Yes.
So in your case, maybe if you have 1%, at normal temperature, it won't really cause an issue. But if you have, say, your chiller breaks down, it goes to 27, 28, it could trigger on their virulence, and then it might start attacking things. The good thing about your tank is that there is a good balance of bacteria, and they definitely compete. So there's some bacteria that will actually inhibit pathogenic bacteria, secrete compounds that suppress Vibrio growth. So having them in the tank in sort of, I would say, baseline level or background level is normal.
Yes.
If it starts going up, then you should probably do something about it.
So this is another one of those ones. It's good to have a baseline value of where it is now, and then if I do another test in 6 months, 12 months time, I can see if it's increased or changed or decreased or whatever it may be, and that gives me some data to track and to work with.
Yeah. So, for example, your water conditions might be good. ICP is good, but then you have some either the tissue necrosis in the corals, or polyps are not extending, or some other thing, and then we see this Vibrio at higher level than background. That could be the culprit.
Sure.
If you want, I can show you what it looks like when Vibrio is when Vibrio is bad.
Okay.
This was from one of a customer had a sick fish, and we'll send them a swab kit. Yeah, this is a level of an active infection. Fish are not happy, and this is opportunistic. It's an opportunistic Vibrio. So something triggered it to become pathogenic. So the virulence factors were triggered, potentially by the conditions of the water and the imbalanced microbiome, which I can show you here.
Oh wow! Yeah.
Okay, so this
it's very different from my table.
It is very different, but this is from the swab. This is the direct swab from a fish. The previous one that we saw was the fish swab. This is the tank that the fish came from.
Right.
We can see Rhodobacteriaceae just dominating the tank, and also high levels of the SOKP, the archaea I was mentioning before. And when we look at the Vibrio, we do see a background level. But yet the fish are showing symptoms.
Yes.
So it is very dynamic. We have a cutoff of 1%, but it's very specific to the strain. It's very specific to the tank conditions and the conditions of the water.
Right.
I suspect that this is why we flag it even at low levels.
Yeah.
There was another pathogen found on the fish that wasn't Vibrio. The Tenobaculum is also a fish pathogen.
Okay.
And I suspect what happened here is Vibrio was a secondary infection to that or other stressful event that they went through.
Right. Yeah.
But then it just dominated the fish's skin once established.
Yes.
Again, you have necrotic tissue. Vibrios love that as a food source, but it's an opportunistic bacteria. It shouldn't do that under normal conditions, and yet we do see symptoms.
Mm-hmm.
And we also find a known fish pathogen that is known to cause that. Put it together, we have a secondary infection from Vibrio that just overtook the microbial population.
Yes, yes. Good to know. Well, it definitely makes me feel a bit better about my values.
Yeah, so your tank definitely looks good. Nothing really stood out, or nothing was really major, except for as I mentioned, the lack of Pelagibacteriaceae. But then it explains it because you're using UV.
Yes.
Is that correct? You're using UV?
Correct, yeah.
Are you using UV as a prophylactic, or if you have an active
No, just
parasite you're trying to treat?
yeah, just as maintenance, not trying to get on top of anything, just trying to keep everything at bay.
Okay. As long as it's working for you, just continue.
Yeah.
Continue using that.
That sounds good. When I scroll through to some of the detail in the what we know and the relevance to your situation, there's some good information in there that sort of summarizes everything that's on this biome report or so far, and it goes through some of the things that I could do. And actually, where does it go through what I could do? I'm sure I saw it there a second ago. Yeah, slightly elevated organics or DOC can increase fibro, so especially above 26 degrees. So then it talks about here we go. Some of the action. Yeah, so I can reduce the Vibrio in the display tank with some granular activated carbon, wet protein skimming. Adding the UV, so continue running the UV, siphoning detritus or improving the flow in some dead spots just to get rid of its food source there, and ensuring that I keep the tank, particularly this time of year, below 26 degrees. I mean, any time of the year, but this time of year is when it's a little bit more difficult. So trying to keep it below 26, and even if possible, as low as 24, which is, definitely something that could be done. And then you've got some other options here to support immunity, to raise dissolved oxygen, keep the temperature stable, obviously feed high quality vitamin-rich food. And then there's some extra options here if symptoms appear around quarantining or hospital tanks or broad spectrum antibiotic baths. So quite an arsenal of options there to me, from some things that I can incorporate into my daily reef keeping through to some sort of steps 3 and 4 there. If things get out of hand, or if I do see things take over, that this is the sort of level we need to go to try and help that.
I just want to clarify that we don't want to just be using antibiotics left and right.
No.
I think it's only for last resort.
Yes.
And it needs to be used responsibly, because we don't want to have giving the wrong treatment and then produce a strain that is resistant to this antibiotic.
Yes, exactly.
So only use antibiotics as a last resort. So pretty much the explanation before, the siphoning, reduce the organic flow, the carbon, it helps to reduce the food source for Vibrio.
Yes.
And just try to dial down the population and hopefully revert it from the virulent stage to the non-virulent.
Sounds good. Yeah. As you say, the last thing I really want to be doing is running a antibiotic. Anyway, as you say, it can cause all sorts of other issues, but it's also it's a drastic measure. So definitely a last resort.
And I think we also mentioned that if you decide to go that route, you do it in a quarantine tank. You do not want to put antibiotics into your main tank.
No.
Because it will definitely change your microbiome.
Yeah.
And we don't want that, because it takes time for it to actually build up and stabilize.
Exactly. Yeah, you're effectively starting your tank as a brand new tank with all of that biome's gone. It effectively wipes it out or clears the slate, and you start again, and you can build up that diversity.
That's right. So it wouldn't wipe out everything, but it will antibiotics target particular, so your gram-positive and gram-negative bacteria, and these species that are more susceptible. So if you wipe out one family, then a different family can overtake and then cause issues.
Of course. Yeah, of course. Yeah, yeah. Really takes that balance out of it.
Yeah, and then it's take your time to get back to your diversity and balance. So antibiotics,
Yes.
not in the main tank, only last resort in a quarantine tank with, of course, advice of a aquatic veterinarian.
Yeah, perfect. Moving on to the next section there, which is around the biofilter. And we get some information here around ammonia oxidizers, nitrite oxidizers, aerobic and anaerobic denitrifiers there. The sort of overview there is that my aquarium is within range. Anything you see there that you think was... I do note there's a little note there saying my aquarium was slightly elevated levels of nitrite oxidizers. Is that anything that I should be
No. So,
paying any sort of extra attention to?
definitely not. And the increase was 0.03% difference. This is more of a range.
Yeah.
It's easier to understand things if there's a range.
Yes, yeah.
This is such a small difference that it's not an issue.
Nothing to wave any flags.
It's
Yeah.
not a flag. Again, when we have more data, we might say, hey, the range is actually
Sure.
0.2% or 0.3. Currently, from what we see and the research that we've been doing, that's the typical range that we see.
Yeah, yeah, makes sense. Good to
Again,
know.
it could have been that day you fed the tank heavily and you sampled it a few hours later. So then, those bacteria population maybe increased just before. It's very dynamic. Such a small difference, I would not really
Yep.
care too much for.
No, fair enough. On the flip side of that, we move on over to cyanobacteria. And I've got a total read of 1.24%. And you do have a note there to say that often over 1% is early sign of a bloom, which I definitely would agree with. I think I touched on before that I've had some little appearances of some cyano or potentially dino flagellates show up before. Nothing's taken hold, but there has been little signs that thankfully with some flow and some nutrients and things have been able to sort of knock on the head, but clearly not knock on the head because they still exist in the aquarium there. They're just not taking hold right now.
Yeah, so that's pretty much what we see in your results. So you borderline visible.
Yeah.
And I see you have one main or one dominant, the Xeniococcus, and also the one that I can't pronounce.
So it's not just me.
No. So this is a benthic one, so you would see it on surfaces.
Yes, okay.
So you have about 10 different types in the system. As you can see, a lot of them are in the background, just doing their thing. They're not really overpopulated. You even have one from the spirulina family.
Okay.
The one that we eat,
yes,
spirulina, the supplement. So they're also found in marine environments.
Yes.
What I don't see, and I think it's probably because of your UV sterilizer, is a cyanobacteria called Xeniococcus.
Okay.
It's a free-floating cyanobacteria. It's a pelagic one. It's not a benthic one.
Yes.
And that's actually one, we think corals can actually feed off it.
Right.
So corals feed off this Xeniococcus. I would like to do some experiments where I culture it and then dose that into my tank.
Yeah.
And see how they do. But I'm basing that only on the fact that when you look at natural reefs, it is a dominant cyanobacteria.
Yeah. That makes
which
sense.
Is not found here.
And it's one of those things where as more of this information is collected, added to the database, combined with some of the effects in the aquariums, you're going to learn this information and develop it, and it could just unlock that next big thing. So super interesting. Heading on down to waste degraders. Now, there's a long list here, but I've got maybe not such a... I've got 3 major types there, maybe 4 minor types, and then the rest are all near zero, if not zero. Anything I should be worried about there? There's a note there to say slight increases are common in nutrient-rich or heavily stocked systems. However, values are above 10% often indicate excessive organic matter or overfeeding. What percentage am I at there? I'm at 20% total. So
Yep.
probably need to give the tank a bit of a move some power heads around, get some flow through there, get detritus up in the water column and filtered out. Is that what you're thinking?
So, yes. So if you have dead spots, if you have a sand bed that collects detritus, if your
Yes.
sump has accumulated detritus, if you feed a lot,
Yes.
if you have a heavily stocked tank. Now, these values, so although we set it to 10%,
Yes.
which is what we see from the current data, that is a typical value. If your tank runs fine at 20, you don't change anything.
Yeah. Okay. Yeah.
If things are going okay, there is no point of changing it.
Yes.
Biology is very, very again, it's very dynamic. There's always outliers.
Of course.
If it's happy, no need to act.
It's an interesting point. It leans heavily on the chemistry side of we talked about ICP and things before, where you might talk to Isan from Triton, and he might sort of say, look, we suggest a value of this for boron, but your tank's double that value and it's doing well. Don't change it. Don't chase the number. Things are happy. Leave it alone. But it's still good to have that number and see if it changes over time, if it continues to increase, or if you see a problem when it increases, or if it was just an outlier and it decreases next time. It's good to know that data rather than act with a bit of a knee-jerk reaction.
How are your nutrients level at? Like the nitrates and phosphates?
Yeah, my nitrates are currently fairly low. They sit around 1 to 2 ppm, which I'm trying to bring up a little. My phosphates are probably not in balance, or they're not in balance with that. My phosphates sit at around 0.12, 0.15 ppm. So it's not quite in the balance where I'd like it, but I am trying to bring that nitrate up.
So I think, yes, this is why you potentially are having a bit of a battle with cyanobacteria. Again, it's a competition.
Yes.
When they have the ability to fix nitrogen, so atmospheric nitrogen,
Yes.
as a nitrate source. So when your tank is depleted from nitrates, or have low nitrates, their competitors cannot grow, but they can because they have the enzymes to convert nitrogen gas into nitrate, and then they overtake. So again, I don't think the ratios is something we should hold on to or give it too much thought, because again, it's anecdotal.
Sure.
We don't really have, not that I've seen at least, concrete evidence that 1 to 100 is a good level, or 1 to 50, or
sure.
In terms of the chemistry of the tank, your tank would potentially do better with higher nitrates.
Definitely something I can target. I can work to increase or reduce the nitrate removers from the tank, if not increase the nitrate in the tank. So
you mean you could remove your algae from the refugium, so dilute the refugium?
I could, or I could take some biomedia out of. I've got 3 large containers of biomedia that seem to be pretty good at targeting nitrates. I could remove some of that. I could reduce the light intensity or time on the refugium.
Yeah. So the media, is that Purigen style material?
It's a ceramic media. So they're hard ceramic balls, just a surface area. Yeah.
So it's a biomedia.
Yes.
Not necessarily an absorber. Okay.
Yeah.
So something that we skipped over, if you go back to the biofilter.
Yes.
So you see the aerobic denitrifiers?
Yes.
I'm kind of skip through them. These are the bacteria that consume nitrates and then release it back as nitrogen gas.
Right.
So you do have a population here, but then I wouldn't say they're overly abundant.
Okay.
They're not extreme. So at least in a bacteria point of view, they're not the ones that are heavily consuming your nitrates.
Okay, good to know.
Essentially, if you dose nitrates, their number would grow accordingly.
Yes.
They would compete with microalgae, with bacteria, with the macroalgae. But some of the animals, like the corals, also are able to uptake some nitrates. So higher values would potentially be beneficial. You might not even see a spike if you dose, because everyone wants to.
They're all going to take a slice. Yeah. Interesting to know. So yeah, I guess all right. Well, with that base in mind, then probably best not to remove the bacteria housing media. Probably more to target some of the competition, like reducing the light intensity or time frame on the refugium. See if that gives the others an opportunity to get a bit more of that nitrate before it bottoms out. Hopefully stop it from bottoming out.
You can even dose nitrates if you do it responsibly and not go too high. Do it slowly. You can definitely benefit your tank.
Yeah. I am currently dosing some nitrates, and as you say, it's just one of those things that you just creep up on, because it can go from very low levels to you sort of hit that tipping point, and it can get away from you quite quickly. So doing very small amounts at the moment, but with some testing, I'll creep up on that and get a bit more in there.
Do you know how much you are dosing per day or per time?
All I know at the moment is I'm doing 3 drops a day. What that value of ppm is for the volume of my tank, I don't know. I'm doing it a little less calculated than that. I'm just going with a small amount first and testing, see if I get results, and then I'll do a little bit more for the week after that test, get some results, and creep up on it that way.
I see.
Coming back down, we get to a section here which I am interested in around biofilm and EPS formers. This is an interesting one, because I've got one type there which is a pseudo something.
Pseudo-Alteromonas.
That's the one. That seems to be fairly dominant, and the notes there say that my aquarium's slightly above typical range, which typical range is 0.1 to 5%. To me, that seems like a big window, and then mine's a fair way above that. I'm up at 8.23%. What sort of effects is this going to give me in my aquarium?
Okay, so that's number one, that biofilms. Very crucial part of the microbiome. They pretty much is a matrix of different microbes, creating a net of polysaccharides, proteins, and they live in this protected niche. Very important, very common. So the normal range is up to 5%. Yours is 8.3. Is it significantly higher? Probably not.
Okay.
It shows me that it's Pseudo-Alteromonas is the dominant one.
Yes. Yeah, that's almost all of us.
They are a good bacterial family.
Okay.
They do produce a lot of bioactive compounds to fight other bacteria, so I wouldn't be too worried about them. Do you remember how I was telling you that part of having a diverse community is having a healthy competition? There's some bacteria that can produce compound that would suppress Vibrio, for example, or keep it in check.
Yes.
So this bacterial family is one of them.
Great.
You would tend to find some tanks in the sump where there's lower flow. You can definitely see the biofilm.
Right.
Where rub your hand against the glass or against surfaces, you can actually see the film. This is the biofilm.
Yes.
So some tanks I've seen has 60% bacteria that are associated with biofilms, which is not. Doesn't represent a healthy microbiome. So your tank having 8%.
It's not a massive red flag.
Perfectly healthy level of biofilm related bacteria.
Good. It's definitely not a sentence I thought I would be hearing this week, but I'm glad to hear it nonetheless. That's good.
In terms of the biofilm formers, so for example, this shows the genus of the bacteria.
Yes.
We don't show it here just to not overload the data.
Yes.
But you could have, say, 20 different species that belong to this genus.
Right. Wow.
Yeah. So this just shows a group of them.
Yeah.
Linked into one name, or
Incredible.
that are linked to the same genus. But yeah. You don't have an overabundance, although it's outside the range. It's still fine. If you have overabundance, you would see your pipes getting slimy.
Right. Okay.
Your equipment, especially in low flow area, would get. And the issue with having high biofilms, again, we want to have everything in proper levels or in normal levels.
Of course, yeah. You don't want it competing with other things.
Yeah, so thick biofilm would create a different environment underneath it where you can have more anaerobic bacteria. It can even trap or provide a niche for pathogenic bacteria to hide under. So if a tank has high biofilm formers, then it would also, I would flag that as well. For your tank is just slightly above range.
Okay, good. I'm happy with that. I'm content. Moving on to pollutant degraders. I'm happy to say that this one looks like my aquarium is within the typical range and in less than 0.1%. Nothing to be too concerned with there. Obviously, it's all within range. There's a bit of everything's doing what it should be doing. Nothing I need to act on or make any sort of changes with.
So in this particular category, we've taken known bacteria that can degrade pollutants. It can be oils, it can be hydrocarbons, environmental pollutants. Say you use a lot of sprays or aerosols in your house for air freshener.
Right.
It would be introduced into the tank. There would be compounds in there that certain bacteria will eat. If their number is higher than the typical range, it may be a sign that some toxins or environmental pollutants are getting into the system.
Okay.
And it's something to flag the user.
Definitely. Definitely worth letting them know before things get, potentially they already are an issue, but even if they're not out of hand yet, best to let them know before it does get out of hand and they can identify what the source is and then stop it from continuing. Yeah.
If there is a pollutant, it might not necessarily kill the animals immediately.
Yes.
A lot of it accumulates and it takes time to have an effect.
Yeah.
So some fish may be more tolerant to it. Some corals will be more sensitive to some pollutants. But this allows us to see if I have any potential pollutants entering my tank or in my house that I should be watching out for.
That's valuable information. And then we come through to sulfur cycling, which is an interesting one. So we've got sulfate reducers and then sulfur oxidizers. My tank is showing there is that its sulfur cycle is balanced, and that there's low risk for hydrogen sulfide buildup. That's a good outcome.
Yes. So sulfur is a very important element in the reef tank.
Yes.
If there are bacteria that cycle it and recycle it. One of the issues there's some bacteria that reduce it into hydrogen sulfide or other compounds. Hydrogen sulfide is just one that is toxic. And the sulfur oxidizers that they take this hydrogen sulfide and use it, convert it to different forms of sulfur, and it's a cycle. Usually in tanks that have more anaerobic conditions, for example, a deep sand bed, low flow areas, we tend to see more anaerobic bacteria like the sulfur reducers. and then you have a higher chance of having hydrogen sulfide production, and it is a toxin for fish and for the other invertebrates. So this just gives us a very easy way to see the balance of these microbial populations. If you had higher sulfate reducers, it would be flagged, and you would will let you know that you need to increase your flow and potentially reduce your anaerobic zones in the tank.
Yep. No, good to know. Thankfully, not something I have to tackle. Something where I might need to do something, though, is some algae interactions. And we've got, although if I'm interpreting this table right, the table there that identifies things that promote algae growth is very, very low. And then I've got some things there that inhibit algae growth that appear to have decent numbers there. However, it does, the little footnote there does say that the algae interaction is dominated by algae inhibitors, suggesting that my system might have low nuisance algae. Is that correct?
Yes. So again, the idea is to take all the data that we have of 50,000 bacterial reads and try to and bring it and get the most amount of value out of it. One of the things that we wanted to show is whether the population of bacteria promotes or inhibits algae, because there's definitely an interaction there. So we went to the literature. We see which populations are associated with, for example, algae disease or algae degradation. Some of them prevent the attachment of the spores of algae, and therefore prevent them from taking hold. The ones that promote algae tend to secrete compounds that promote their growth, growth regulators, nutrient exchange. So based on this, your microbiome doesn't, or the specific one that interact with algae doesn't favor their growth.
Okay.
This is what we get from this figure.
Good information. That then leads through to a nice summary here of the entire results from the Aquarium microbiome summary, which is good. And it gives just a nice little way to sort of encapsulate all of that data we went through, because to be fair, particularly on this microbiome test, there's a lot of data there to take in, and I'm fortunate enough to have you on the call with me here going through some of these things. But for other people, they would obviously have a look at the results and take in some of these sort of footnotes for each of the graphs and data they see there, but they can also have a look at the summary here. You and your team, though, are very good at, as you've touched on a few times today, when there is something that is a level that is high, and for the average user, if I see a value that's 0.1% or 5%, to me that might not mean a lot, but you guys do know, and that's when you've got your knowledge through the database and through your experience, and that's when you do flag it and say, we think this is a problem, and you make sure that the end user knows through the report, but also potentially through any of these flags, or even through like the emails back that they get the results from. You're going to let them know that there's some things that they need to take action on, or at least be aware of.
Yep. And again, as I tell the customers, when they get the reports, if they have any questions, feel free to message us, because we're happy to help.
Yes.
We want them to succeed with their tank. And we are getting a lot of our customers that are asking questions. What does this mean? What does this species mean?
Yes.
And sometimes we can guide them through their problems.
That's fantastic.
We go through it together.
Fantastic. Well, it's two incredibly next level reports that just didn't exist in this hobby as little as a year or two ago, and you've come along with ReefLabs, made them available to the hobbyists here in Australia. It's fantastic, and both of these tests are now available through the ReefLabs website. That people can go to reeflabs.com.au, have a look at the shop there. You've got the microbiome test, you've got the P3 test, or what I'm going to push for is the P3 plus test, with the algae in there as well. You've also got some other products that are coming soon, and you've also got a bio scrub, which is it's a type of coral dip as well. So you guys are not resting on just these two incredibly intellectual and really eye-opening test kits that you have. You're branching out into other products as well.
Yes. One of them that we're working on, it's still in beta testing. We try and get more data. Is a way to stabilize your microbiome and push it towards a healthy one if you're deficient in some bacteria. And that's where the biometric gel comes in.
Yes.
We design it so it looks like it's just a gel in a little container.
Look at that. There's the product right there.
Yeah, still on the testing. Some people are already testing it in their tank. We already know that it supports a very particular type of bacteria because it has food for specialist ones. It doesn't feed any pathogens.
Right.
But yeah, it's still early. Hopefully we'll get it out soon. And then we also have in the pipeline, an immune boosting food for fish.
Right. Yes. Okay. That's what the recovery boost is here. I'm sharing the screen now for those watching along at home. Interesting. Yeah. Good to see. I must admit, on first glance, I thought this was a type of carbon, but no, this is a food.
Yeah, so it's still a long process to show the evidence because we're doing trials, and eventually we have to do challenge trials. To actually prove that it boosts their immune function. But we have some specialized ingredients in there that are proven to induce different parts of the immune system and make them more resilient. So we are trying to provide more health-related products for the reef industry, or reefing community. We have all sorts of crazy ideas, but we really have to focus on, a few things at a time.
Of course.
We do have a few things in the pipeline.
Very exciting to see. Well, I want to thank you once again for taking the time, one, to test my aquarium, both on the P3 test kit, but also on the microbiome test kit. Taking the time to go through this call with us here and share your insights into some of the results of my aquarium and break it down a little bit further for those who may be looking to do either or both of these tests, and they'll get their results and go through similar things. They'll be able to see my results now and compare with them and have a look at what you commented on those. And of course, you're available via message or email when end users do get their results. If they do have questions, they can go back and forth with yourself and the team and get more information on the health of their aquarium, which is an incredibly tailored service, which I think is very, very commendable. So well done.
No worries. Thank you for having me. I'm glad to go over your results with you. I hope the viewers got value, you got value out of it. And if anyone has a question, either related or not related to this, feel free to reach out. Some people reach out through our Facebook page.
Yes.
We're always happy to engage with the community, get their feedback, and just help them have better thriving reefs.
Fantastic. And likewise, if people watching the video have questions that they want to see a follow-up video on, they want us to dive deeper into a particular aspects that we didn't on this video, feel free to let us know in the comment section below. We can always jump on another call, record it, and share that information with the public out there. But at this point in time, my brain is about to explode. I'm super pleased with the results. I've got some things that I can work on, some things that I can look to improve in my aquarium, some things that I know are fine for now, but I'll track those values over time. And it's, like I said, it's just a space that we didn't really have access to in this hobby in such a short time frame ago. So to have people like yourself making this possible really takes this hobby to the next level. So thank you once again, and keep up the great work.
Thank you. Appreciate it.
All right, guys, there you have it. That is the deep dive into the ReefLabs P3 and Microbiome test kits. An absolute evolution of testing on our reef tanks. Something that I never thought would be possible, in my lifetime at least, is to run a DNA sample across all things in my aquarium. It absolutely blows my mind. And the wealth of information that is provided from those two test kits just is still sinking in all of these many minutes later, still getting my head around what that means for my tank and what I should do next. But I'm super, super grateful to have Dr. Tom on the phone like I did today to go through those results and explain what it means for my aquarium. And I totally recognize that's not a standard part of the service. However, I do want you to rest assured that if you do purchase either or both of these test kits, you do get that full report which explains all of the information, and you also get follow-up email support from the ReefLabs team if you do have any questions about those values. So there's a lot to take in, I completely understand that, but it is an absolute step forward in our reef aquariums. And just to see what some of the bacteria, what some of the pathogens, pests, parasites, some of the other things that are happening in our tanks, the different diversity of different bacterias and things going in there, really is something that we're just starting to scratch the surface of. And I can say that the ReefLabs team themselves are continually evolving this process even further. In the time alone from recording this video and going live, there has been some addition. And Dr. Tom wanted me to tell you about a new section in the microbiome test kit that looks at some phosphate assimilating bacterias, which is just another evolution again, which is super, super exciting. Now, Dr. Tom was also very generous and said that any of our Parker's Reef viewers that do go ahead and purchase either a P3 DNA test kit or the microbiome test kit will receive a free ReefLabs Bio Scrub, this fantastic coral dip designed specifically for SBS, although it can be used on other corals as well, but specifically on those sensitive SBS to eliminate problems before they get into your reef tank, will be included free of charge whenever you purchase any one of those test kits. If you want to have a look at this product, the test kits, or anything else from ReefLabs, be sure to go to reeflabs.com.au. I'll have all the links down below so you can check out their products there. And of course, support local and help us evolve this hobby even further here in Australia. I do want to point out, if you have any questions, comments, feedback for myself or for Dr. Tom on anything to do with the ReefLabs test kits or products, feel free to pop it in the comment section down below. And if there is anything you'd like to see covered in further detail, please let me know, because Dr. Tom was very, very willing to do another session answering any of the questions you have, which I think is incredible, and I would love to take advantage of that. So please don't be shy, ask your questions below. I'll answer them there if I can, but if not, we'll get Dr. Tom back on another phone call. I also need to give a massive shout out to these names on screen here. These are my channel members. These guys chip in a couple of dollars each and every month to make it possible for me to do crazy things like this, DNA testing my dream reef tank, and without their support, it would not be possible. So thank you very much to these names here once again. Last but not least, if you're yet to subscribe to the channel, please consider doing so. It takes 2 seconds of your time, does not cost a single cent, but it does support the channel, and I'd personally appreciate it very much. Anyway, guys, I look forward to seeing you next weekend with more new reefing content. Till then, stay safe, keep reefing. Cheers, bye.